Hi, everybody. Welcome to our next unit on population genetics and speciation. Um, so, if you decide to become a biology major and you go to the university, you'll have to take a genetics course, and it's the weed out course for the bio department. It's pretty tough. It's a lot of math, and up until this point, I haven't asked you guys to do any math, really, more or less, as far as I can recollect, but we'll do a little in population genetics, right? We study populations and we measure populations, and we're going to do a little bit of that in a few seconds. So we call population genetics specifically microevolution. All right, so we can look at changes in small groups and come to conclusions about how they're evolving. Of course, how we measure that variations in groups is an important concept we'll explore. And then, of course, remember, we accept Darwin's theory of evolution through natural selection. And remember, that change is based on mutations. All right. And so we'll look a little bit about how mutations arise and how that gives rise to variations in populations. Well, microevolution is and population genetics are ways of looking at specifically the change in the types and numbers of alleles. All right, so remember that an allele was a different copy of the gene. We'll use this eye color bogus example just because it's very simplistic. And remember, we said that there's two alleles for eye color, brown or blue. We know that's not true, but we're going to go with that just for this simplicity's sake. And so we can look at these changes in the brown allele or blue-eyed allele in populations and make assumptions about what's happening at a genetic level, how a population might be evolving. Remember that Charles Darwin was the father of genetics, but he didn't know anything about genes. I'm sorry, I'm sorry that I made a mistake. Mendel was the father of genetics. Darwin was the father of modern evolution. But he didn't know anything about genes. And, remember that the gold standard for really understanding biology at this point is biochemistry. And so we'll look at changes. We've looked a little bit about how changes in genes affects us. And we'll examine that again. So microevolution is population genetics. And so we increase our understanding of evolution through genetics, and that evidence supports the theory of evolution. And of course, we're learning all kinds of cool stuff about genetics as we go forward. And the link between microevolution, right, population genetics, and then evolution generally is described as speciations. All right, so we can tell how we get new species arising. Of course, if you look around, you see all kinds of phenotypic variation. You and your brother or sister are different. You're somewhat like your parents. But if you look around, there's all kinds of variation in populations. And population biologists can look at individuals and measure some specific phenotypic trait, and then describe how that population might be changing, how that might be undergoing speciations. Of course, we're all the same species here on planet Earth, but we come in a lot of different shapes and forms. So, phenotypic variation. Remember, your phenotype are the traits that you have, and we can look at these observable traits and measure them. Okay. And remember, we've talked about the bell curve in the past. It's also called the normal distribution. It's a mathematical term. It's normal because the mean, the median, and the mode are all equivalent. And there's this classic bell-shaped pattern that emerges. And if you look at this curve, you see that 72% of the population will be found between the first two quartiles around the median and mean. I know that that's complicated. That's statistics talking, but we'll find 72% of the population under that first big part of the curve. Now, there's another concept in statistics when you do a statistical study called standard deviation. Okay, so the mean, the median, mode are all the same in the normal distribution. If I go one standard deviation forward and behind, I'll find 72% of that population. In the second quartiles, we only find around 11%. And then beyond the second quartiles, we see about 3% of the population. And we use this normal distribution to grade you in, right? About 3% of people should get A's, about 11% should get B's, 72% of you guys should get C's, 11% should get D's, and 3% should get F's, but we don't do that anymore. Everybody gets a B. So the bell curve is really important in understanding populations. If a population is under ideal circumstances, no matter what you measure, you'll see something similar to a bell curve, whether I measure your IQ, or I measure the size of your feet, or the color blueness of your eyes. We end up seeing sort of normal distributions. So remember that Darwin contended that the source of variation in individual was mutation. And they have to be mutations that can be passed down. So they have to be mutations in germ cells, in the sperm or the egg. So remember, many mutations are silent. They don't have an effect. And then the other common mutations, or missense or nonsense, they have a bad effect, right? They don't have a good effect. And so mutations that are actually adaptive, that is, they help you, are really rare, and so they get passed down at a very slow rate because they don't help. Most mutations aren't going to help you reproduce. It's only these rare mutations that are adaptive, and so let's talk about measuring those changes, okay, and how to measure the different alleles that we see. So we will look at allele frequencies, right? And so let's go to our eye color example again, an allele frequency. So we're going to say you have a brown eye allele and a big B, it's dominant, and the blue eye allele, a little B. I can look at how many people have those traits and give you some mathematical answers as to how frequent they are. And by changing frequencies of alleles, whether it's the eye color changes or the spots in your coat changes, we can determine speciation. We can measure speciation and the evolution of different populations. All right, so what's the allele frequency for the little b eye color gene in your class, the blue eye color gene in your class? So let's do this. Hang on. Remember that we use this simple model, the brown eye allele, blue eye allele. So we have three genotypes: big B, the homozygous dominant, big B, the heterozygote, or little b, and that gives us only two phenotypes, right? You either have blue eyes or brown eyes, and big B gives you brown eyes, little b gives you blue eyes, and the heterozygote big B, gives you brown eyes as well. So if we just said that we had 20 students, right? That means they have 40 eye color genes, right? We get two of each. Let's say that 9 of the students have blue eyes and 11 have brown eyes. Well, what's the allele frequency of the blue eye gene? Well, I know that since there's 9 of them, that means they have 18 blue-eyed genes, the blue-eyed persons. I know what the blue-eyed person's genotype is because it's the homozygous recessive, right? If you have brown eyes, though, I don't really know what your genotype is, right? You could be big B, you could be big B, little b. And there's a way to figure that out, and it's called the Hardy-Weinberg equations. And so let's take a look at that. Um, Hardy-Weinberg is a central idea to population genetics. Um, it's a measure of allele frequencies, um, and, but then there's some of the other important ways that genetic changes can occur. Right, sexual reproduction is a source of variation. Right, we mix our genes, we shuffle genes during prophase one of meiosis. Right, and then with organisms, population size really does matter. Okay, it really affects how rapidly changes can occur. So I have population size. Even the environment's going to have really important potential changes on populations, and so we'll look at these patterns that emerge. Well, Hardy-Weinberg is a idealized version of populations. And it's idealized in the sense that it doesn't really exist. And it doesn't really exist because you have to have these very strict terms that under which it operates. And we'll look at those in detail. But first, let's take a look at the mathematics. Now, the things that can't happen in a Hardy-Weinberg population are gene flow, non-random mating, genetic drift mutation. But we'll talk about that in detail later. Let's look at the math. So there are two equations. First equation is p squared plus 2pq and q squared equals 1. Second equation is p and q equal 1. All right. And what this relates to is our genotypes. Okay. For example, here's the eye color problem. So the question was, if I've got 9 blue-eyed individuals, how many alleles do they have? Well, we said that the allele frequency for that would be 18 of 40 or 0.45. All right? And it could be 9 to 20. I'm still going to get 0.45, right? What that represents is this q squared term. All right, so here we go. P squared plus 2pq plus, does it look familiar? Well, this really represents big B squared and 2 times big B little b and b squared, or big B, 2 times big B little b, and little b. That's what the equation means. I know this because I can identify the blue-eyed individuals. In fact, little b represents 0.45% of that population. Now, in order to find little b itself, just a little b, I have to take the square root of 0.45. That gives me 67, which means that the rate of, or the allele frequency of little b is 0.67. Now going to this other equation, p plus q equals 1, or big B plus little b equals 1. I just substitute that in, and that means big B's allele frequency is 0.33. Now I can go and substitute these values into the equation, or I can just take 2pq. 2 times little b times big B gives you 0.44, and that's the frequency of the heterozygote. Let's do another question. Sickle cell anemia occurs in 25 of 10,000 individuals. So how many carriers are there in a population, right? So I know little s, right? It's 25 of 10,000. To find s itself, I take the square root of that, 0.00025, right? The square root of 0.0025 is 0.05. If I know little s, I know big S, right? Big S is going to be 0.95, little s is 0.05, and then to evaluate for this carrier question, I take 2 times P times Q, and that gives me 0.95. So the frequency of carriers is 0.95. You may ask, like, who cares? But, well, look, that's how we can take measures in microevolution. We can look at allele frequencies and determine what kind of changes are happening in populations. We'll come back to Hardy-Weinberg next, and I'll be giving you guys a couple little math problems to solve. It's not that hard.